For me, the ocean means everything. I grew up in the French Alps, in a mountain region tucked between the high peaks of Mont Blanc and Lake Léman. But ever since I grew up, I've been fascinated by the ocean. I've been diving from a very young age, spending time in the Mediterranean and learning about just how fragile the marine ecosystems are. And so since then, I started my career in marine science and now work as a communications expert and now a non-profit director. I left Geneva when I was in my late 20s to go to Scotland to study marine science. And so that's when I finally realized that this was going to be my career path, my journey as a marine scientist, but also as a filmmaker and consultant, that the ocean was going to be a major part of my life and that I was going to work towards its protection, its restoration, but also communicating its importance, which is the work that I'm doing now. So in March 2020, I was in Aotearoa, Christchurch. I decided to start a non-profit foundation and give it a name. The COVID pandemic meant that I was grounded, that my international work was not happening, so I decided to start something local at the community level, and I named it Blue Cradle. So when I think of non-profit organizations working in marine science and conservation, there are many names out there and all were taken, except one. I thought, well, blue naturally is the color of the ocean, but also thinking of the terms cradle and how we come from the ocean, and also that we need to cradle the ocean. So those two words together, Blue Cradle, mean a lot to me, and they mean a lot to a lot of people, and I feel that it was a great name that actually encompasses the responsibility that we must take to protect the ocean for future generations. So in June 2021, we set out with our inaugural Blue Cradle expedition. We left Tamaki Makaurau, Auckland, and we sailed all the way up to the Bay of Islands on board Manawa Nui, led by Jochen Zeichmar. Jochen is a marine mammal specialist and he knows a lot about the region, and so we went with him because we knew that we were in good hands.
Aotearoa Gulf, I think, is New Zealand's largest harbour, is it? It's a big embayment, very shallow, in most places sort of shallower than 50 meters, and it's a fantastic, amazing ecosystem with lots of different islands, habitats, home for lots of species, and under a lot of threat, lots of impacts from fishing, farming, boating, you name it. Well, so monitoring is really important in the Aotearoa Gulf, or anywhere really, because there is a lot of wildlife. It's a very important ecosystem for New Zealand, and a unique one at that. And it's just really important to also get that baseline data so you can, really understand trends and changes and react accordingly and adjust your management. It's an amazing ecosystem that really deserves greater protection.
Ora mai, ora mai. Ora mai ki te taua nei. Nau mai, haere mai, whakatau mai. I mua i ngā mimi. Me tāhuri tātou ki te kaihanga. Nāna nei ngā mea katoa. For those who are going on the expedition tomorrow, big mihi to those that are going on the vessel. Tangaroa e te titi, tangaroa e te tata. Tangaroa up high and below. E o ake ranga te rangi. Clear the angry skies. Kia tai te wakaoru ki uta ki te enua. So that the famous Voyager Ru's canoe can land on the island. Te ua e te matangi. The rain and the wind. Akarongo. Listen. Marino, Marino. Be calm. So hopefully you guys will have a calm voyage.
So this is my first time on the Manafanui. Just, you know, witnessing the beauty of this boat just now. Yeah, it's a really exciting time. So we've been unloading packs and packs of material. Yeah, and I'm tired. I need a beer. I think that sums it up, right?
Yeah, for sure. Yeah.
So shoes, we're supposed to remove the shoes? Or?
The shoes have a good
Thank you very much. Thank you. But everything you leave outside sort of goes with the night.
My name is Anastasia Zaiko. I'm a co-lead of the Marine Biosecurity Toolbox program, and I'm a researcher at the Cochrane Institute and also a co-appointee at the University of Auckland. We got together with Blue Cradle and another really nationally important research program, the microplastics in New Zealand, AIMS2. And we decided that if we join our forces and work together, we'll be able to organize a research expedition and collect data on these two major problems in New Zealand, the biological invasions of non-indigenous species and also the microplastic spread in New Zealand marine environment.
Before that, I can say that the elastic bands that are there, inspired by Scandinavian kindergartens, make your own color combination on your glass, and then I don't have to wash the glass absolutely all the time, because if you hide it somewhere, and I will go get it, I will just grab it and wash it. So put it there, put it out in the sink when you think that now is a good time to have it washed.
I'm good, but we're still overexcited with loading off.
Very good.
Great.
Oh yeah.
Such a good idea.
So I need that. Yeah.
It's a conversation starter, and it says a lot about your personality.
What does that mean about me?
I'm Dr. Olga Pantos. I'm a senior scientist at the Institute for Environmental Science and Research, based here in Christchurch. I'm the co-lead of a 5-year MB-funded project, which is called Aotearoa Impacts and Mitigation of Microplastics. And so the project's looking at the levels of microplastics in New Zealand, what types of plastics they are, the impacts they have, and also looking at some of the solutions.
We're living in a critical time right now with the impacts of climate change on the world's ocean. We've got sea level rise, ocean acidification, deoxygenation, all these things happening at the same time. And on top of that, we've got the anthropogenic pressures that we're putting on the ocean, such as pollution, plastic pollution, overfishing. And so really the ocean is at a tipping point. So the work that we do at Blue Cradle is about collecting data, bringing scientists together, going out there in the ocean. And then that data is then analyzed, processed into the lab, and then that information is then combined and condensed into scientific papers and journals and reports that then go towards the policymakers. And so the policy decisions that we make are well informed. We've got the best available science informing to then produce the best available policy to then have a benefit on our society. So that's what we're trying to do. We're trying to enable that process to happen and to facilitate those expeditions.
Day one today of our expedition. We're on board the Manawa Nui, and we're here with a whole bunch of people. So from Blue Cradle, Alga Leader, ESR, Cochrane, and together we're doing a whole bunch of different sampling, focusing on plastics and biosecurity.
So the sampling that we did in the trip that we took from Auckland up to Northland, it's work that's not been done before, so we really don't have any idea of the levels of plastic, or if there are areas of higher concentrations than others as well. And of course these environments are critical for a lot of marine species. So we need to better understand the relationship between the land and the water, because there's a disconnect, because things may not be quite so obvious to everybody. They may think there isn't a problem, but if we can have a look and see and show that there are actually plastics within these marine environments, we can better protect them.
Okay. We use a manta trawl, which is a device that was designed to target surface water samples for microplastics specifically. And so it's a modified plankton trawl that sits with its wings, so like a manta ray. It can glide along the surface, and its big open mouth as a filter feeder, like the manta ray. It tows behind our boat to capture everything that's on the sea surface, and we process that in our lab to quantify the amount of plastics that are on the sea surface, so that we can have an understanding of what the impact might be to the ecosystem here.
I'm Hayden, and my role here is part of the microplastics collection team, where we have the manta trawl out the back collecting surface microplastics. So once we get our manta trawl in, we've done the filtering, and then we wrap our filter up, as you can see in here. So we've got one filter wrapped up here. And the reason we use aluminium is because we don't want to use anything plastic, because that can contaminate the sample, and then things such as paper and that, when you're in an environment like this, just gets wet and soggy, and it's just a disaster. Our different samples in here, just all labelled up, ready to take back to the lab, where we'll do some possibly digesting of the plankton and then density separation of the microplastics so they float to the top, and then we can pour that off, look under the microscope, and analyse for how many microplastics there are and what type of polymer there is, and that's our results. So I'll see you guys there.
My name is Xavier Pochon, and I am a marine molecular ecologist working here at the Aotearoa Institute for 10 years now, and I'm also an associate professor at the University of Auckland. So the Marine Biosecurity Toolbox program is a nationally significant program that involves 16 different institutions, both here in New Zealand as well as abroad, which is a really amazing program that has multiple components, all focusing around the problematic of marine pests. Here in New Zealand, we've got a long evolution of organisms that have adapted in isolation through the rest of the world. So the ecosystem are stable and everything is in place. Now, when new organisms travel or find their way through to New Zealand, the majority of those will be very well adapted and quite resistant to new environment, which means that they can come in this new ecosystem, really outcompete, be quite aggressive in some cases, and outcompete fauna and flora that are established here. And what that creates is an instability of the system where they can take over, use resources, and displace the sort of diversity that we're used to here. Traditional plankton net is typically something like this, but the problem is that if you go faster than 2 knots, invariably the little parachute's gonna go at the surface and go crazy. And so when we did this initiative, Plankton Planet, with sailors going around the world in the open ocean trying to collect plankton for us, well, that was a bit of a problem. You know, they hated that, of course. 2 knots is just impossible in the high seas. And so this is why we came up with this thing. So there's no rocket science here, but still costed about $20,000 to get to this thing, believe it or not.
You're basically like able to sample with that like at much higher speeds than with the traditional net that we're used to. So we can actually go up to 7 knots with that net now, which is pretty cool because it enables like much more bowls to sample with it. And
it kind of looks like a toilet seat, no?
Yeah. That's what everybody says.
It looks like lamprey mouse.
Yeah, like a lamprey, you know the fish, the docks on. So like basically the water flies in here and then it accumulates plankton at the end and the cod end. And then in the end we're gonna take the cod end with the accumulated plankton in here.
Yeah, but also we have modified version here, so we can insert filters straight into this thing and collect sample immediately while sampling.
And then I just go to 500, yeah? Boom! What Anna got a great idea about, that's quite brilliant. She came up with this idea of modifying the code ends. At the end, you've got this section where you can actually insert some filters directly in there. And so what we're essentially doing here, we're testing all 3 scenarios. The normal code ends with the cruising speed net, the modified code end, so the liquid that remains on top, so we filter that, and then the filter only. And so we're really interested to know whether we recover the same genetic biodiversity in these 3 different scenarios. And if we do, then that's a game changer because we don't need to do this whole filtering system. What we really want to do is to simplify the whole process so it takes less time, it's easier, and it gets the answer that we need.
Beauty, eh? So this fin would be much taller if it wasn't so wobbly. Oh, hello!
Oh, hello!
It was a friendly slap.
Yeah?
Hello, hello, hello.
Oh, he's big.
Yeah, wee! Fantastic, that.
It's quite shallow in this bay.
Yeah.
Crazy.
Away!
It's amazing.
Good.
Got that array there.
This is so... Oh!
See it? Oh.
Ah, yes. Yeah. It's for your website. Our 2 expeditions, they form sort of the foundation, the building block of Blue Cradle as as an entity, as an organization. As we go on into ocean literacy education.
Right now we've got some Year 7 and 8 students from Whangarei Intermediate to come out onto the boat, and they're learning about the plastics issues we have in the ocean. So they're looking at macroplastics and then how they break down into tiny little particles, making microplastics.
Say you put a pot of, I was just going to say water.
I was thinking the same thing.
I think it just gives so much more understanding to the issue, and also hopefully can give them some inspiration as to what they can do about the issue. So
We're starting to develop curriculums and working with schools, and we really consider that the relationship that we have with these scientific institutions and these senior scientists is really fundamental, because we've got this understanding of the work that they're doing, the importance of it, but also how it needs to be communicated and taught.
We turned the Earth into a giant game of Battleship.
So hopefully they'll be inspired and represent that next generation of scientists and environmental activists, and to help protect, look after our environment and the moana.
We just had
140 school
Really?
children
It was
on this
140?
boat over the space of 4 hours.
No wonder you needed that.
Just teaching them about microplastics and how ocean gyres work and some of the biosecurity stuff that we're working on, and communicating the science and how it works and what we're here for. Hopefully having some impact around that.
So we are going, heading to Nayland College. For me as a scientist, it's, you know, I can either stay at my lab or I can do a bit more and interact with kids and share some of this knowledge that we're developing. And I get a lot more through that. If you can communicate to kids, chances are you'll be able to communicate to everyone.
They can become real pests in New Zealand's environment. They can establish their population, they can compete with native species, they can even outcompete native, valuable populations from the environment.
One of the main goals that we've done through the program is to try to incorporate this knowledge into the curriculum, like build a package that they can use year after year, and so that you can actually really measure changes, and that's what we're trying to do today.
So in New Zealand, we've got a really unique opportunity to define our own ocean literacy curriculum and incorporating other things like citizen science and working at the coastal level, not necessarily from boats, but at the very local level, at the community level, collecting meaningful data and back up through the institutions to then produce meaningful scientific papers and reports, which can then have an influence on the policy change.
Security is one aspect to the whole health of New Zealand.
It's beautiful, we've never been here before, so I'm just going to be up here the whole time. We're going to do it all over again on a different boat.
So we're in the Milford Sound, which looks like something straight out of Jurassic Park, but even better because it's real. We're about to head out into the open ocean and we're on our second expedition. So we were up in Northland back in June and now we've come down to Fiordland and we're back at it with the microplastics and biosecurity data collection. You can see our surroundings. We're so remote compared to when we were up in Northland. We're always off the coast of kind of human activity. So being so remote, hopefully there'll be less microplastics here. But yeah, we'll see. It's kind of hard to believe that there would be pollution in somewhere like this. You feel so far removed from, you know, human activity in your everyday life. This boat is called Stranick. It's captained by Rodney Russ, who has graciously invited us on board to do our research.
Okay, 2 knots, deploy now.
Tool deployed now.
It's the most epic boat. It's stunning. I think we're very lucky to be able to call it home for the next week.
Look at that, it's too easy.
In the Fiordland, our main goal was to test the setup of a small onboard molecular laboratory to make sure that the methods, the complex of different techniques can be used outside a true molecular laboratory settings. So we managed to do that. So we even ran sequencing on board. We had a few suspicious samples where we suspected that there could be Undaria DNA present, but then we confirmed it wasn't Undaria DNA, it was like probably another kelp species that we detected. So it's a good news for the Southland Council, because we didn't find Undaria in those areas that they didn't expect to have it. And in the same time, we also used these samples to understand the biodiversity of the region. We are still analyzing this data. We have some preliminary results, but not the final yet.
Well, I've lived and played and worked in here for a few years, yeah.
What's so special about it?
All the fjords are special. This is just one of the many special places in the fjords, yeah. Yeah, I mean, the end of the track is good. There's a lot of history up there and some of the named places. Henry Saddle, Mount Henry's all named after Richard Henry, one of New Zealand's earliest conservationists. Yeah, so no, it's just good.
We've just been processing all these samples, so each one of these is part of the trill that we did from each site. There's heaps of stuff on there, and you just got to sort through it all, visually identify what could be plastic and pull it out and then analyze it. So it's this bright yellow, fragmenty film looking thing. No wee fragment here. I'll focus on that. This is all from Fiordland. Once we get it onto these filter papers, the first stage is this here, so looking under the microscope, pulling out any of the plastic. We have this disc here, which is a diamond cell, and then from there we're able to pick the plastic off, and then we can compress it, which just makes it as thin as possible, so more light intensity can get through when we scan it. I'm looking at these surface trills is that we are getting more variation than a lot of these other things we looked at, like mussels, which is you'd expect. Definitely a lot more in there so far.
I do buy food in plastic. I avoid it where I can, but go and get the loose stuff from the shops. But that's not accessible to everybody. There's so many items that you just can't avoid it. It's not easy and it's not equitable. But that's where policy legislation needs to come in. Because we don't need everybody to be an expert on plastic packaging.
It took us a while to analyze these samples. We collected quite a few. So from the Northland expedition, our primary goal was to test some of the new devices that we developed for better collection of environmental DNA and compare them with the older devices. So this comparison was really, really successful. So we could show that the information that we collect with both devices are comparable, and our new way of collecting samples is very efficient, but also produces the same level of information. We actually detected some non-indigenous species in those samples, and in the next few months we will use some other analytical methods to define the spread and model the populations around the area.
What he's doing right now is taking every single sequence read and compares it to this dedicated, fully annotated reference database. Here we are, boom, so that's the first output, so it looks like this. So
tells us that it found 20 native matches with marine non-indigenous species, and these are indicated in this purple color. We do not have any unwanted and notifiable organism found. That is good.
That's a good news.
But if we would, there would be a red flag against these species.
So now the next step is really to look at the abundance of these different species and the proportion of those species that are actually not yet known here in New Zealand. So there's a great chance that a portion of them will be possibly new to science, but that will require the appropriate research behind it to figure out.
So usually we see like hundreds and thousands of species detected in our data sets, and I would say up to 30% of those sequences cannot be identified to a species level, or even sometimes more, because we do not know our biodiversity well enough And that's really important to discover what is in our waters, to make sure that whatever arrives, it can be detected effectively.
And so part of this problem is that here in New Zealand, there's, this genetic analysis of the environment is fairly recent. It's only a few years that we've been focusing on that. And so we're greatly lacking all of this genetic information of the biodiverse habitats here in New Zealand. This is really essential, but it needs to be done properly.
Yeah, to make sure that our biodiversity is well captured in these gene bank database, for example.
So the journey continues.
So what we've got in Aotearoa New Zealand is unique, is we've got leading world scientists working on some of the solutions to restore the ocean's health, but we also have ancient knowledge passed on from generations found in the Te Ao Māori worldview, personification of nature, integrated, holistic, ecosystem-based management. And with those two worldviews working side by side, we can provide solutions to the rest of the world. And so this laboratory that Aotearoa New Zealand is right now is potentially one of the best solutions that we have to address some of our greatest challenges around the world. So when I think about the future, I feel hopeful, but also realistic. I think we have an opportunity right now to change the course of history. We can listen to the signs from nature, we can listen to the different worldviews that we have, or we can ignore and continue down the path of consumption and capitalism and destruction of the natural world that we live in and that we rely upon. We started this mahi, this work, with Blue Cradle, and we now have partnerships with iwi, we have partnerships with communities. We are working side by side to figure out the best solutions, not for the next 2 or 3 years, but for the next 100 years. And that requires bold, ambitious investment right now in our communities, in our local communities, in citizen science, investing in the solutions for tomorrow. So nature-based solutions, working with the best available science, the best available Mātauranga Māori knowledge, and side by side on the dual hull waka, we can then find a pathway for the future, and then the rest of the world can watch and listen and learn and hopefully replicate.